Hello, everyone. Welcome back. I'm Pauline Assis. I'm joining you guys all the way from São Paulo, Brazil. So it's night for me now. And I'm assistant professor at the University of São Paulo in Brazil, and I'm a member of the diversity committee in the SSE. So now I would like to welcome our next speaker, Dr. Pratibha Sanjenbam. Pratibha is a postdoc at the National Center for Biological Science. Her work aims to understand the key ecological and evolutionary processes which govern the interaction of beneficial microbes with their host plants. She will speak today on how host landrace shapes the phyllosphere microbiome. So welcome, Pratibha. I'm looking forward to you.
Thank you, Pauline. Let me share my screen. Hello, everyone. I'm Pratibha Sanjenbam from Dr. Deepa Agash's lab at National Center for Biological Sciences, India. And I'm really excited today to share some of my recent findings on host microbial communities. We all know that most eukaryotes harbor diverse and numerous microbes in their bodies, and this serves as a good model system to understand the ecology and evolution of to understand the ecology and evolution of microbial communities. Wait, it's got stuck. Sorry. Yeah. Sorry for the technical glitch. Yeah, but in today's talk, I will be preferably specifically focused on plant microbe association. And when I say this, many people in the audience would have thought about the microbes which are associated in below ground, which is the rhizosphere associated microbes. A large body of work has already illustrated the establishment of microbes and the assembly and the impact of these rhizospheric associated microbes on the plant processes. How similarly, the aerial part of the plant is also home to various microbes, and these microbes can have huge impact on the plant fitness, thus establishing setting up the possibility of evolution between the host and the microbe. Hence, many recent studies have now actually started to focus on understanding the assembly and maintenance of stability of these phyllosphere microbial communities. This brings me to this basic microbial ecology question that what determines the distribution and diversity of phyllosphere bacterial communities in nature. To help address this, I chose a fantastic model system, which are called rice land races. These are nothing but the local rice varieties, which are originated and evolved for millions of generations in a specific niche, selected for certain traits like for higher yields, aroma, or grain colors, etc., by the local farmers during rice domestication. And in many reports, it has been shown that they possess high phenotypic and high genetic diversity, setting up the possibility of imposing divergent selection on their bacterial associates. And indeed, in my prior work where I isolated a specific bacteria, which is shown in the picture over here, these are called Methylobacterium, and they were extracted from the rice leaf surface. And indeed, in my experiment where I showed that having Methylobacterium with its own host landrace could impact or have tremendous beneficial impact either in the greenhouse experiment or in the native field site. However, this impact was not generalizable, suggesting the importance to identify the host and the evolved partners of the host bacterium pairs in nature. That's why I'm interested in understanding the factors governing the distribution and diversity of phyllosphere communities, which are associated with these land races, basically aiming at bacterial communities to begin with. Existing on the prior literature and my prior work, these are some of the expectations of the microbial communities that I expect to see in these land races. Broadly, the Phyllosphere communities can be divided into two groups. One, bacterial lifestyles which are adhering on the leaf surface, I will term as epiphytes, and the bacteria which has the ability to stay in the leaf tissue as the endophytes. And we have environment which is consistently changing over time. So what do we expect? Very basic, right? We might expect to see very higher richness and diversity in case of epiphytic community as compared to the endophytic community. And we might expect to see a weaker effect of host landrace since they are exposed to the environment all the time, the epiphytic communities, as compared to the endophytic community, where I expect to see stronger effect of the host rice landrace since they are in close proximity with the host tissue. And in fact, I would expect that less stable community in the case of epiphytic microbial communities over time as compared to the endophytic community. To test this out, I carried out an experiment. I sampled leaf from this rice land races at a particular stage, which is called the flowering stage. And it was collected from a specific location as shown over here. It's called Manipur, and it lies in the northeastern region of India. And these are the sampling years that I have collected. I collected consecutively for 3 years, which is 2019, 2020, and 2021. And here are the number of land races which I collected across the years. But today in this talk, I will be focusing on only these 6 consistent rice land races for my further analysis, which are consecutively sampled across the 3 years. And here are the number of plant replicates that are collected per land race across the years. And these were all collected in the field. And as a control, I also included a very new introduced rice variety, which are called high yielding variety as my control. So once the samples are collected, it needs to be transported back to the lab as soon as possible. Then the DNA are extracted. So firstly, we go for the extractions of DNA from the epiphytic community, and the same leaf has been used for the endophytic DNA extraction. So once the DNA is extracted, I proceed for microbiome analysis using 16S rRNA V3V4 amplicon sequencing. And once the sequencing is done, I generate an assigned taxonomy of the sequence DNA using data2 pipeline, and different sequences in this pipeline will be referred to as amplicon sequence variant, which I'm going to call as ASVs. In my most of the slides that I'm going to talk today will be in terms of the genus level and in terms of the ASVs level. So what do I mean by that? For example, if 3 ASVs are identified and they belong to a specific or similar genus, they are going to be clubbed together as one specific genus. And that's what I'm going to call as genus level analysis. And if I take the assigned ASVs for my further microbiome analysis, that will be the ASVs level analysis. For instance, to make very simple to understand, let's take a scenario where I pick a very distinct rice landrace, which is called black rice, as shown over here. The grain colors are really black, and they are locally known as chakao. And here are the taxa which is color coded over here. There are some dominant taxas which I'm highlighting over here, and the rare taxa which are here are the bacterial reeds, which are less than 1%, are then clubbed together to form as the rare taxa. So what do we find? So on the x-axis, I have the number of plants that are being sampled, and y-axis is the relative abundance. It seems like in epiphyte, we have more taxa than compared to the endophyte. But the idea to show it here is that to highlight over here is that there is very little variation to begin with, individual variation to begin with for further analysis. Is that true for the remaining years? So I took another year, and it is quite consistent over for a given landrace and for a given time. And if I take another landrace to compare with, it is quite consistent even for other landraces, suggesting that similar processes might be responsible for the community structure in these landraces. Given this result, I further proceeded to test out my expectations, which I already set earlier in my earlier slides, right? So what do we find? We find that, say, for example, the community as I coded over here, the blue color is my endophytes and the gray color represents the epiphytic community. X-axis will show the landrace and Y-axis will show the Shannon diversity index. And as expected, for example, in 2019, we see that the epiphytic community tends to be more diverse than the endophytic community, but they are not statistically significant all the time. But the pattern seems to be true. That is, the epiphytic community seems to be more diverse than the endophytic community. Is that true for the remaining years? And that's what we see. The similar pattern is consistent over time as well. And we do expect this basic pattern to be true for that high yielding variety that we included. And that was true for that also. Suggesting that the epiphytic community, which is exposed to the environment, will have more richness and diversity than the endophytic communities. And this was similar when we did the pattern analysis for the richness, which is the number of ASVs, where we find epiphytic community to be much greater than the endophytic community. Given
Excuse me,
this result,
Pratibha, 3
yeah.
minutes, okay?
3 minutes. Okay. Given this effect, we wanted to ask whether we have weaker host effect on the epiphytic and community as well as in the endophytic community. And surprisingly, we see that if you check the R-square value that's shown over here, that most of the variation, even in the epiphytic community, is explained by the host landrace, suggesting that host imposed selection might be irresponsible for structuring the community. And if we combine all this, we still that there is strong effect of landrace than the year, which suggests that even in the environmental fluctuation, landrace seems to be the strongest predictor in this phyllosphere communities. But the other way to test out is to ask if the proportion of rare taxa are much higher. Test out is to calculate the rare taxa, which is a proxy to measure for the stability, is to compare the rare taxa in the endophytic and the epiphytic community. Let's take an example over here. We see that the pattern seems to be true, where the rare taxa proportions are higher in the epiphytic community and compared to the endophytic community. And this was generally true then across all the land races. However, the pairs was not statistically significant all the time. And in order to test out, a very simple test is to calculate to carry out a network analysis. So what we do is we take all the samples and then take the relative abundance data set and convert it into their correlation matrix, calculating the pairwise correlation of each taxa. So we visualize it. For example, the nodes here will represent each taxon, which will presented over here is the genus 1, genus 2, and genus 3. And if the ages will represent what kind of relationship that taxa has with genus 2 or 3, which is indicated here. And the more connection the nodes has, the degree of the nodes will be bigger and darker in color, right? And we see that a complex network exists in epiphytic community than the endophyte communities, right? And given this variation, I then wanted to identify the core members which are relatively selected by plant for their beneficial effect on their plant fitness. So I then asked that given all these samples, what are the core consistent members which are present as an epiphytic group as well as an endophytic group? So here on the x-axis, I have the relative abundance data, which is given a detection threshold from 1% to 10%. And on the y-axis, we have the each taxa which is present across these relative abundances, and the prevalence is calculated by the presence absence, which is shown in the color code over here. For instance, let's take an example. This Metallobacterium is prevalent across all the samples, and it is present in higher abundant in the community. Whereas if you take Stenotropomonas, it is hardly present in any of the relative abundance. It's only present in 1% and in very, very less sample. Similarly, we find this in terms of the endophytic community. However, to highlight that there are bacteria which can live both epiphytic as well as endophytic lifestyles, as for example, Metallobacterium. There are specific genus like Pentoa, which loves to be an epiphytic lifestyle, Aeromonas, which seems to love the endophytic lifestyle. To summarize, very low individual and temporal variation with strong effect we see in border communities. Epiphytic tends to be more diverse with stronger impact on host landrace. Higher modality and complex network in epiphytic community was observed, which suggests that the stability of this community is going to be less when compared to the endophytic community. Despite this variation, we see that the core genera are still identified, and many new open questions are now coming up, whether the community composition has impact on the plant fitness, and what is the mechanism of interaction between the bacterial symbionts and the host-rise plant. With this, I would like to thank my mentor, Deepa. I'm really grateful to her to giving me this opportunity to work on this project. Nitish, my colleague, who had helped me with some of my codings. Basically, he's my code partner. And my father, who had really helped me in sampling samples during pandemic. Without him, I wouldn't be presenting some of this data set today. And of course, my fantastic lab members, former and current lab members, and also the farmers, which I really thank, specifically Sharad and Ibobi, who are generous to give me samples all the time. The next generation sequencing team here, lab support, kitchen, and of course, the funding sources. Thank you, and I'll be happy to take questions.
Thank you very much, Pratibha. Fascinating talk.
Very slow. Oh, yeah, thank you so much.
We already have one question here, so I'm going to read it for you. So epiphytes are less stable than endophytes, as you said. Does this not contribute in lowering the richness of epiphytes, though you have shown that epiphytes are more diverse than endophytes?
Yeah. Yes, it's true that epiphytes are predicted to be less stable over time. But the richness diversity has already been shown that the diversity tends to be higher in the epiphyte. That itself already shows that there is new arrival of species, and there is a chances of the stochastic events to happen. Right. So I did not quite understand. Does this did not contribute in lowering the richness of the epiphytes? Yes, we might expect to see it, but we don't see that. We see that even though the landrace was explaining the highest variation in the epiphytic community as well, the richness or the presence of the rare taxa is making the epiphytic community less stable than the endophytic community. Did I answer it clearly, or?
I guess so. We have one more question, quick question, and then we have to move on, okay?
Yeah.
So thanks for the interesting talk. On the conclusion slide, could you please show where the bacteria are exactly? I'm interested in their morphology.
I didn't quite get it, but I'll try to answer this. Where the bacteria are exactly, does that mean the surface bacteria or the endophytic bacteria? I'm not getting that. So I mean, on surface, as I have shown you over, they reside near mostly near the stomatal site. I can quickly show.
Oh, in the image you showed.
Yes, so they generally reside there.
Okay.
Sorry, it got stuck. I'll just explain to you. For the epiphytic bacteria, we generally see near the stomatal sites where they are found in higher abundances, and also near the trichomes where we see these hairy-like structures. I already have data on it, but for the endophytic bacteria, according to the existing literature, they might be residing near the membrane and next to the cell wall. So I'm not very sure about the endophytic cells because yeah, I don't have data on that. Or I mean, I'm not clear what she meant by the whether it was epiphyte or endophyte in the first case.
Okay. Yeah, thank you so much, Pratibha.